Thank you all for coming today. Thank you to ACMP and the program committee for selecting me. Today I'll be showing you some of my postdoctoral work looking at the effects of the interleukin-1 system and the role it may play in alcohol dependence. So the neuroimmune system is well known for its role in the brain's neuroinflammatory response to things like infection or injury. But it's becoming clearer and clearer that the neuroimmune system and its signaling pathways can also play a role in basal cellular function and in more complex behaviors like learning and memory or social play. In addition, dysregulation of these neuroimmune elements may also play a role in several neuropsychiatric disorders, such as addiction, schizophrenia, depression. Specifically, my lab, the Marissa Roberto's laboratory at the Scripps Research Institute, is focused on alcohol use disorders. And it has been shown that alcohol and the neuroimmune system have very complex interactions. So, sorry. I don't know what I just did there, sorry. I don't know what happened there. Oh, there we go. Is this the pointer? Yeah. So, sorry about that. And so, it's been shown that chronic alcohol exposure causes changes in many different cytokines in the brains of alcoholics. And in addition, these neuroimmune pathways can cause changes in synaptic activity in many addiction-related brain regions, such as the hippocampus, the cortex, the central amygdala. And also, these neuroimmune signaling pathways can be manipulated to affect alcohol-related behaviors, such as alcohol intake or alcohol preference. So this has led to the general idea that perhaps with each exposure of alcohol, we're seeing changes in some of these inflammatory responses in the brain. And over time, these responses can accumulate, and as a result of this accumulation, this can push the brain into transitioning into alcohol dependence. In particular, the interleukin-1 system is considered to be a key regulator of the brain's response to alcohol. And this system is a pro-inflammatory system that's quite complicated, as you can see. But today, I'll be focusing specifically on one particular part of this system. So I'll be focusing on, I'm sorry, I can't get this pointer to work. I'll be focusing on IL-1 beta, the agonist, and the IL-1 receptor. And the reason I'm focusing on this system is because it's been particularly implicated in alcohol use disorders. Chronic alcohol-dependent people have been shown post-mortem to have high levels of IL-1 beta expression in the cortex and other brain regions. This has also been replicated in ethanol-dependent rats. IL-1 beta exogenous application to rodent models has been shown to increase alcohol-induced neuroinflammation. It promotes withdrawal-induced anxiety, and it leads to deficits in cognitive function. In addition, the IL-1 beta system has been shown to regulate inhibitory transmission in several addiction-related regions, as I mentioned. However, despite the fact that there are these indications that IL-1 beta might be involved in the cognitive function and the cognitive deficits of alcohol dependence, no work has looked at IL-1 beta's function in the cortex. And this led us to the question of, does alcohol affect IL-1 regulation of cortical inhibition to impair cognitive function? For our studies, we used a chronic intermittent ethanol two-bottle choice model of alcohol dependence. And for this model, we induced ethanol dependence by exposing C57 Black 6 male mice to chronic intermittent ethanol vapor sessions that are interspersed by two-bottle choice ethanol water sessions. And this protocol has been shown to generate alcohol dependence and physical dependence in the mice. This protocol has 3 different treatment groups. So the alcohol-dependent mice are shown to escalate their drinking, and in their final week of two-bottle choice sessions, consumed about 4.5 grams per kilogram of ethanol per session, while the non-dependent animals that don't receive the alcohol vapor, only the two-bottle choice drinking, drank a much lower level of 2.8 grams per kilogram, and we term these animals non-dependents. Finally, there's a third group, the air control group, that receives no alcohol exposure throughout the entire paradigm. And after this 10-week procedure, the animals are sacrificed after their last vapor session, and the tissue sliced for electrophysiology recordings. So for this study, we specifically recorded from the pyramidal neurons in layer 2-3 of the prelimbic cortex, of the medial prefrontal cortex, and we selected this region because the prelimbic cortex is known to be involved in drug-seeking behavior. And we have previously shown that these exact neurons, these pyramidal neurons in layer 2-3, are particularly alcohol sensitive. When we applied IL-1 beta directly onto our slice, doing our slice electrophysiology experiments, we found that IL-1 beta decreased the inhibitory transmission onto the prelimbic layer 2-3 pyramidal neurons, as you can see on the left. Interestingly, we were interested in how IL-1 beta interacted with alcohol. So we applied acute alcohol onto our slice and then applied IL-1 beta on top. And we found that with the interaction of acute alcohol, IL-1 beta now had the exact opposite effect. Instead of decreasing inhibition of these pyramidal neurons, it instead increased inhibition of these pyramidal neurons. And this was a pretty distinct finding. We were excited by this finding, and we wondered whether this effect was long-term or transient. So we did a second experiment where we applied ethanol directly onto the slice, we washed it off, we applied IL-1 beta on top of the slice after the washout, and we found that IL-1 beta returned to its normal effect. So this seems to be an example where the interaction of acute alcohol and IL-1 beta causes a transient change in the intracellular cascades that IL-1 beta is recruiting to have its effects on inhibiting these pyramidal neurons. When we looked in the non-dependent animals, we found a similar effect to the control animals, so IL-1 beta decreases GABA transmission onto these neurons. While in the dependent animals, we saw that IL-1 beta increased transmission onto these neurons. And this was particularly striking because in the dark blue bar, there's no alcohol on board. Even though these animals were sacrificed directly out of the vapor chambers, by the time these experiments take place, there's no ethanol in the system. So this seems to be a long-lasting effect. And it suggests that this interaction between IL-1 beta and acute ethanol, over time with these repeated exposures in the dependent model, actually leads to a persistent neuroadaptation in IL-1 signaling. We were interested in trying to understand the mechanisms underlying this persistent change, and the way we did this is by using pharmacological blockers. So the IL-1 system is known to be able to recruit the MyD88 P38 inflammatory pathway. And in addition, only in neurons, and specifically this has thus far only been published in hippocampal neurons, IL-1 beta can also recruit a second pathway, the PI3K AKT pathway. So we wondered if these two pathways may be underlying this mechanistic switch. And to cut a long story short, for reasons of time, we found that this was true. So we found that in the naive animals, IL-1 beta signals through the PI3 AKT pro-survival pathway. And with acute alcohol, we see a shift. And as a result of this shift, IL-1 beta is now signaling through this MyD88 pro-inflammatory pathway. It seems that chronic alcohol exposure solidifies this shift, making the IL-1 system have more permanent neuroadaptation. This led us to the second question of whether this IL-1 beta induced deficit in cortical function is associated with the cognitive impairments that we see in human alcoholics. And to answer this question, we modeled cognitive impairment using the Barnes maze. This is a fairly simple task where the animals are placed on a large circular board with 20 holes drilled into the board, and the board is elevated, so the animals have experienced a direct drop from the board down to the floor. And only in one location is an escape box placed where the animals can climb in. There are spatial cues oriented around the room to allow the animal to identify which direction it's facing and more easily learn where to find its escape hole. So we trained our animals in this task, and after training them, we put them once again into this chronic intermittent ethanol 2-bottle choice vapor paradigm. And after the 10 weeks of exposure, 3 days into withdrawal, we tested our animals for their retention of this behavioral task. And what we found is that the alcohol-dependent animals actually showed deficits in this task. 3 days into withdrawal, they had a longer latency to escape, and they showed more errors in terms of trying to find the hole before they successfully found it. And just to show you a couple of videos of what this looks like, this is a naive animal, And once the cup is lifted, the hole is directed right here to the left. And once the cup is lifted, the animal runs directly to the target quadrant and pretty quickly finds the hole of interest. This is an example of a dependent animal, and this time this mouse's hole is actually directly on the right, where the blue arrow is. And you can see the animal seems to essentially be running randomly around the board looking for the hole. So the animal knows the task. It knows that it needs to find a hole to escape into, but it is having difficulty identifying where that hole is. And it's important to remember that when these animals were trained, these two animals performed exactly the same. So this is a clear deficit that was caused by the exposure paradigm. And as you can see, it takes a little while, but it does get there. But another important thing to note is that I ran velocity testing on all these groups of animals, and there was no difference in the speed at which they ran the task, indicating that they're performing equally in terms of their locomotor abilities. So he eventually finds the hole. One thing that I alluded to just now while we were watching the videos is there can be different strategies that the animals use to find these holes. And one particular strategy is the spatial strategy, which the first animal performed, where the cage was lifted, it looked around, it spatially oriented itself, and then it ran directly to the correct target quadrant. Another possibility is sequential, where an animal will run directly to a hole and then start sequentially checking each hole to see if that's the correct hole. And a third possibility, which is what we saw of the dependent animal, is random, where it seems to just try its luck. And we found that with the chronic alcohol exposure, there was a distinct shift in the different strategies the animals were using on test day. So there was a large group of animals that started to use a random strategy to find their hole and escape the task. So overall, what I've shown you today is that chronic alcohol exposure seems to cause persistent neuroadaptation in the IL-1 beta signaling system, leading to changes in inhibition of cortical neurons. And I haven't shown you this, but I have shown you, but hope to show in the near future, currently running the experiments, that these changes in IL-1 signaling may be linked to cognitive function in terms of deficits in spatial memory. And I think this is really exciting and really important because we're starting to move away from this idea that psychiatric disease is just linked to general neuroinflammation. And instead, if we identify these specific neuroimmune pathways, we can then think about druggable targets. And for example, the IL-1 receptor antagonist is already used as an FDA-approved drug to treat rheumatoid arthritis. And so the idea of repurposing can be really exciting for the alcohol use disorder field. So with that, I'd like to thank my mentors and my labs, particularly the Marissa Roberto lab and Amanda Roberts, who's helped me a lot with the behavior. And I'd like to thank my funding, particularly from NIAAA, where I recently received the Pathway to Independence Award. Thank you.
We have a few minutes for questions. Randy?
So your data on the plasticity of the IL-1 response, very interesting. And I wondered what, I think you summarized that data. I'm not sure you actually told us the data that supported it. So was it biochemical, staining?
Oh, sure. Sure, so it's been primarily electrophysiology data up until now. Scott Edwards' laboratory at LSU has been doing some molecular work looking at phosphorylation of some of the kinases, and that's currently being done. But so far, it's been primarily electrophysiology.
So when you looked at this AKT-PI3K switch to this MYD P38, that was done by staining?
No, so that was primarily done by using the electrophysiology and using particular antagonists to block different effects. Okay. Yeah.
All right.
Great, thank you.